right, let's go ahead and get started to keep things on schedule. So I'm very excited to welcome you to session 3, which is on the emergence of large-scale neural architecture. Our first speaker is going to be Matthias Kaschube, who is a professor of computational neuroscience and computational vision and a senior fellow at the Frankfurt Institute for Advanced Studies. So I'll go ahead and hand it over to him.
Yeah, thanks a lot for the introduction, and thanks a lot, Ilya, for setting up this great symposium and for inviting me. I think it's really a very broad and interesting set of talks so far. So my work is at the interface between neurobiology, computer science, and physics. And what I'm going to talk today is largely inspired by a work together with Gordon Smith, who was just talking after me. And so these are basically 2 talks back to back. And so what I'm interested in a lot in this context is how neural representations arise during development. And for instance, think of the representation of the external visual world, right? It starts out this process starts out way before birth and continues, of course, after birth. And often it is thought about in terms of nature versus nurture. So you have, on the one hand, you have the endogenous mechanisms, and you have experience-driven mechanisms. And the endogenous mechanisms are thought to be most important early in life, while the experience-driven mechanisms become more and more important at later stages. But in the context of this workshop, I want to emphasize really that actually what's also is very important is that there are feedback loops, dynamical feedback loops, on various levels during these processes, right? So as we have seen in previous talks, and as we all know, these structures have to be built in space over time. And so it's highly dynamical processes, and these dynamical processes, they involve a lot of dynamical feedback loops. And so there's strong dynamical loops at virtually any levels. And so there's ample potential for self-organization. And, one of the examples of self-organization would be, for instance, the spontaneous formation of lipid bilayer, right? So you have that possibility to do that, but what a developmental system may want to do is what I would call guided self-organization, right? So basically it uses this ability of, or this possibility that structure can form through this process. It can use this in the right time to build and use it where it's where it's useful to implement the kind of functionality it needs to do. Yeah. So, any idea who this person is? Who this young person is? There's one old friend who is already has a good intuition. Yeah. So this is. Of course, a young, bright mind, still developing his brain, so not having fully matured yet, but at some point will be developing concepts which form the basis of computer science and actually also of a lot of what we're doing, what you're interested here, basically self-organization and development. And so that's Alan Turing. And he's, of course, in his last paper, he introduced what is now known as the Turing mechanism. The Turing mechanism being basically a mechanical or like a dynamical mechanism for generating a structure with a typical spatial scale. So in this case, you have an activator that activates itself and forms like a localized concentration of this activator substance. And then you have another substance, which is an inhibitor. And both diffuse, but the inhibitor substance diffuses more, more rapidly. And so the inhibitor actually suppresses the activator. And so this suppression leads to like a reduced concentration of the activation further away from this peak. And so what you have as a result of this is a blob or bump of this activator formed in the center, which is flanked by basically a suppression of this activator. And then a new, in further away at a distance, the activator again form a bump. And so via this mechanism, of which is essentially local activation and lateral inhibition, you can have a repetitive pattern being formed in space. Yeah? So this is a mechanism that can select dynamically a pattern with a specific spatial scale. And so that is the way that Turing has formulated this problem in his original work. In the context of neuroscience, we think of this mechanism more in terms of what is provided by lateral connections. And so in a simplest toy example that I would like to discuss here on this slide, you can think of excitatory connections from this position in the center at 0, which has a certain lateral spread. So this is excitatory connectivity. And then you have inhibitory connections, which also have a lateral spread, which is further in distance, in x. And then the sum of the two leads to a structure which is sometimes called Mexican head. In this context here, I would like to call it LILI, local excitation, lateral inhibition. Yeah, that's just the sum of the two connectivity profiles here. And the function of it, what it does to neural activity, can be understood best in actually frequency space. So when you take the frequency representation of the excitatory kernel, then you have something localized in space. It becomes very non-localized in frequency space. So this local excitatory connections become, lead to this rather non-localized kernel for the excitatory connectivity. Then you have the broad inhibition leads to something localized in the frequency space. And so when you add the two, right, adding in real space, when you transform this in the Fourier domain, you basically add them in the frequency space. And you get something by adding the two interaction kernels in frequency space that has a specific peak at a finite frequency, right? And this is essentially this peak is where you have most of the growth of the Fourier modes of this frequency, and this is why you get a pattern of a certain frequency or of a certain wavelength, right? So this mechanism gives you a dynamical amplification of a finite spatial frequency and is defining the pattern then of this specific wavelength. This is pictures. This is how it looks like in like a standard modeling framework in circuit neuroscience. This is basically the Wilson-Cohen type of models. You have rate units. You have two populations, excitatory and inhibitory. This is encoding rate. And this is basically, the rate is basically decaying to 0 if there's no input to the network. And the input to the network or to this population is given by this lengthy expression, where this is the input to the entire network. And these are the basically the terms that mediate the interaction between these two populations. So basically how the excitatory population excites itself, how it's excited by the inhibitory population, and the same for the inhibitory population. And so the range of these interactions, these are the MAIs, and they are indicated here. These are Gaussians, right? And so they fall off as a function of distance. And then basically, LILI, or Mexican head, means that the range of excitation is more short range than the range of inhibition. And when you do this, so schematically it looks like this, right? Green is positive, red is negative. You have this LILI connectivity. And as a result, you will get a modular activity pattern. So an activity pattern both in the excitatory and in the inhibitory population where you have specific peaks of activity which form a regular pattern, in this case a hexagonal pattern of these activity blobs. Yeah? And so this has a quite characteristic spatial scale, and this spatial scale is set basically by the range of the lateral connections, yeah? The excitatory and the inhibitory ones. So this is the basic mechanism in these models, and you know, the type of structures that has been observed in the cortex, in particular in the visual cortex, that has inspired many theoreticians to look into these kind of models are, for instance, the so-called orientation preference map. So this is a paradigmatic example for cortical representation. It's the representation of visual edges, so how visual edges that occur in different parts of the visual field are represented by cortical neurons. So basically, you can think of this visual field as being mapped onto the cortical surface. And on the scale of a millimeter, you have different cells preferring different orientations, and it varies mostly gradually as a function of space. So these domains here, for instance, these red domains, hundreds and thousands of cells in this area would encode for the vertical orientation of stimuli in a particular visual field location. And so this intriguing structure in the visual cortex has a very defined spatial periodicity, like a characteristic spatial scale. And so it has inspired many people actually to model the self-organization of such structure with Turing-type, Lilai-type mechanisms. And so this is actually a field of research which dates back more than 50 years now. So I outline here a few studies that have been proposed in the previous decades to explain basically how this structure could emerge via rules of self-organization that involve local excitation and lateral inhibition, including also some of my own work. Some of these papers model activity. Some of the papers actually model directly the relationship between tuning curves, but they have all in common that they share basically this basic dynamical mechanism for generating the modular structure that is visible in this orientation preference map. And so these are all modeling studies, and it has been traditionally one of the major questions whether these models, to what extent actually could be verified empirically. Like how can we actually empirically test the validity of this general approach? And this has been something that I was personally struck since actually the beginning of my career. Actually, in a paper, actually the first paper that I published 20 years ago, I tried to use image analysis methods that were available back then at the time to see whether there's any evidence that the orientation preference map, and here you see an example taken from with an old experimental technique, basically radioactive labeling, whether there's any evidence of a genetic component controlling the structure of this spatial pattern of selectivities here. And so what we observed back then is actually that the periodicity of between columns, so the spacing between columns appears to be genetically controlled, and the evidence for that was that leader mates seem to have turn out to be closer in their column spacing than non-leader mate. Or the data from non-leader mates. So basically, on the left-hand side, you see the different symbols correspond to different leader mates. On the right-hand side, you see a significance test. So within leader mates, basically, the periodicity of the orientation preference columns was more similar. And so also, I should note that this periodicity also varies between different species, between primates and carnivore species. And so the interpretation that emerged here was that model activity is generated by the dynamic Lilai mechanism, yet the parameter of Lilai are influenced by genetic factors. Yeah? So this could be interpreted, or this is a question I put up here, as one possible interpretation, that this is an example of guided self-organization, right? So basically, the Lilai mechanism, this lateral interaction, generates this modular, nearly periodic structure, but then under genetic control, maybe the range of lateral interactions, for instance, by controlling the size of the dendritic arbors or the range of axons. And so basically, by this controlling the parameters that determine at the end the specific spacing, right? But the emergence itself of the pattern is via this self-organization mechanism. And so this brings me to what I would like to get into in this talk today. So Lilai is a very robust and effective mechanism. It's a mechanism that provides a rich structure from a very simple rule, but it has never really been tested in vivo. Yeah, and we have done work on this, and this is work that is later in the next talk presented by Gordon Smith. So I would like to defer to him on this question. What I will discuss here is I want to ask whether Lilai is actually consistent with known cortical anatomy. This has always been a question for several years already, and I want to show some recent work on this, and also address the question of what happens when actually you have heterogeneity. So cortical circuits are not as nicely homogeneous as we often like to model it. And it turns out that heterogeneity in the local connections has actually interesting consequences, and I would like to discuss these consequences. So first I start with Lilai and whether it's consistent with cortical anatomy. So what is the evidence for or against Lilai? There's one highly cited work on the left-hand side, which has sort of the dream experiment, right? You take a patch of the cortex, a slice, and you patch two cells, and you check whether they are connected or not, and then you basically look for the histogram of connectivity as a function of distance. You do this for excitatory cells on the left, on excitatory, inhibitory, and so on. And then you can trace out basically the distance dependence between the lateral interactions. Here it looks like there's not really a difference between E to E, E to I. However, the caveat is this is in mouse auditory cortex. And mouse auditory cortex is a brain structure that is not known to produce actually modular activity. So it may be not surprising that you don't see any evidence here for that. In ferret visual cortex, not the exact same experiment has been done, but some other method, glutamate uncaging, basically uncaging glutamate and looking for activity at a further distance away. And the method is not as precise and accurate. So the data that was obtained here looks like the two distributions are again quite similar. But as I say, yeah, the experiments are not as clean. It's not that they are fully conclusive. So it's still open whether there is evidence in favor or against actually a Lilai mechanism in ferret visual cortex. So this is just saying it's unclear. Yeah. So the spread of local lateral inhibition might not be wider than excitation, but future experiments need to resolve this. What we can do at the moment is ask in terms of a model, do we actually need an anatomical Mexican head? Or is there a way also that we could get an effective Mexican head without actually having a real anatomical Mexican head? And this is something I want to propose here today in this talk. So on the left-hand side is what would be not having an anatomical Mexican head, right? I to E is more short range than actually E to E. And what I want to argue is that with strong local self-inhibition, eye-to-eye self-inhibition, you can actually get what I call an effective Mexican head, where effectively the range of E to I to E, this inhibitory loop, is actually more wider in space than the E to E loop. And the basic argument is actually relatively simple. So when you take the model that I previously described, and I can make this argument easier in terms of the mathematical side of things and less so like giving you the main intuition, but mathematically it's actually quite simple. So when we have local self-inhibition and we plug it in here, then what we get on the right-hand side is actually this term here. The integral basically breaks down, right? And this term is similar actually to that term, just a different prefactor. And so we can combine these two terms and divide by this prefactor. And when we divide by this prefactor, we see that actually the time scale here, now dividing by this common prefactor, becomes smaller than the previous time scale, depending on the strength of self-inhibition. So with increasing strength of self-inhibition, actually the time scale becomes shorter and shorter. And this has interesting consequences. And this consequence has been, the consequence has been studied in previous work of a short time scale. Actually, when it's easiest to see when we go in the limit of very small time constant, then actually this second differential equation here becomes, we can adiabatically eliminate actually the time dependence. And then we have basically this expression here of the inhibitory activation, which we can then directly plug in here. And when we plug it in here, then we have basically this integral and this integral over these two kernels. And these are two Gaussian kernels, which basically becomes a convolution over two Gaussians. And if we compute this, then we get again a Gaussian. Convolution of two Gaussians again gives us a Gaussian. But this Gaussian here then has a sigma, which is the sum, the quadratic sum of the sigmas of the individual Gaussians. So this just means that this Gaussian here now, which is the inhibitory Gaussian, has now a larger spatial width or range than actually the excitatory Gaussian, which means effectively we have a Lila mechanism by this mechanism. And so this shows that by in the extreme with strong local self-inhibition, we effectively can extend, effectively extends in space the lateral inhibition, and we can get an effective Lili even in the regime where anatomically we don't have a Lili. So this is a boundary on the left-hand side. Anatomically we don't have a Lili or a Mexican head interaction. On the right-hand side we have, but with local self-inhibition, actually this regime broadens up, and the parameter regime in which we have an effective Lili becomes larger and larger. And this becomes stronger and stronger the stronger the local self-inhibition is. This is just a linear stability analysis, our phase diagram. Next slide shows numerical solutions that actually in this regime we in fact get in our full nonlinear model model activity in E and I despite the fact that there's a lack of anatomical Mexican head. So bottom line is this strong local self-inhibition, we can increase the parameter regime in which we get an effective Lili interaction, and by this the modular structure as an outcome of the dynamics without having actually an anatomical Mexican head. And so is there evidence for local self-inhibition? Well, there is by classical connectomics work in a way. So this is EM work having investigated inhibitory basket cells in a catfishal cortex and found that there is a strong number, highest number of autopsies. This is one way of how strong local self-inhibition could be generated. Another way is that it could be mediated by local interneuron circuits. So this closes this first part of my talk about how could we can we expect to see model activity despite actually having an anatomical Mexican head. Now I want to shift into the second part, which is about the effect of heterogeneity. Okay, fine. Let's assume there is an effective lateral interaction that generates model activity. So far, the models that I have shown to you, they were showing homogeneous cortical sheets, right? In every location, you have the same interaction kernel, and they form these beautiful hexagonal modular patterns. Now, real cortices, of course, we wouldn't expect that the lateral interaction is so homogeneous and the same in every location. We would expect to see some heterogeneity, and here I just show you, for example, a few dendritic arbors of layer 2, 3 pyramidal neurons, and of course, they look different neuron to neuron, right? And there is some spatial heterogeneity. So there's no really way of expecting that interaction kernels are fully isotropic. And so what is the consequence of this? What changes when Lilai connectivity actually becomes heterogeneous? And so I want to contrast what you get for a homogeneous isotropic connectivity model to a heterogeneous one. The homogeneous one, as I showed you, generates these hexagonal activity patterns, right? This is like activity generated with input, uniform, weak noise. And when you look at the correlation structure, when you pick one domain and look how this domain is correlated with other domains, then actually the correlation structure is isotropic, and the correlation decays as a function of distance with these radial target pattern-like structure, reflecting basically the typical scale of the system. But it's fully isotropic, and it must be because the dynamics is isotropic, right? There's no reason why the correlation structure should not be isotropic. Yeah? Any solution, any activity here pattern would like a shifted or rotated pattern would be an equal solution. Yeah.
How does heterogeneity enter into your equations? So what is the nature of your heterogeneity?
It's basically there are different ways, but one is basically you deform these Lilai interaction kernels. You know? You add some noise. This can be high frequency or bandpass filtered noise. So basically you deform.
What was done in this case?
Yeah, deformed. So a static noise on the interaction kernel. Static, yeah. So here's no plasticity, connectivity doesn't change, right? So it's a static perturbation. And it's different for every kernel. So it depends on the space, basically. And so, but this is still homogeneous, right? When you do heterogeneous, basically the activity patterns, they are much less regular. As you can see, they are not as hexagonal anymore, and the individual domains have become a bit elongated, a bit stripe-like. Now the correlation structure looks very interesting. So first of all, the correlation now is space dependent. So every location has a different unique correlation pattern. And so it's location dependent, and it is much stronger and much more long range. You can see this by comparing basically the different colors here. Yeah? You have stronger blue, stronger red, meaning you have stronger correlations over broader distances. Yeah? And this is really an emergent property of these heterogeneous, strongly coupled networks. So you have strongly coupled functional networks emerging over a distance of several column spacings away. And so these strongly coupled networks, they have interesting functional properties. And one interesting functional property is something we want to test, and this is a hypothesis, is that it actually matters a lot when you think of what drives these networks, what kind of input is actually activating these networks the most, then it really matters whether you hit one of these distributed functional networks or you don't. You know? So this would be a random network, random input, and this would be an input that actually hits these functional networks being these dark black patches, right? And the hypothesis is that this leads to stronger amplification. Yeah, and this is of course an important difference because when you think of these networks emerging early in development, and then you want to drive them with sensory input, right? The sensory input, in order to be effective, it should actually drive those subnetworks that can lead to lots of amplification in these networks. Right? And so we studied this in a network model similar to the one that I showed you before, just now one population model already reduced. Yeah, this is the type of heterogeneity. So here you see it visually. This is a deformed lili, right? Deformation is again different at every spatial location. In order to get at like what are these functional networks, we looked at actually we went into understanding in terms of connectivity. So what are the connectivity modes that would show us how we should drive the network in order to elicit the strongest response? And so since these are non-symmetric networks, we use singular value decomposition. Basically, singular value decomposition to decompose the effective recurrent network. And the idea is that this decomposes this connectivity into a set of input and output modes, each with a singular value. The input mode basically related to the drive of the network, and the output mode to the resulting activity, and the singular value would give us the strength of this transformation. So basically saying that once we know this decomposition and we use an input that is aligned to the leading input mode, then basically, because it has the strongest singular value, it would lead to the maximum possible activation in this network. So by this decomposition, this is how the input and output modes look like, and we can now either construct aligned inputs that is aligned to the leading modes, or random inputs which is distributed over many modes, and we can look at how the normalized singular value looks like as a function of the connectivity components, and we see that depending on where we are, which connectivity component we drive, actually we get very strong or weaker amplification, right? Just basically given here by the singular value. This is all still in the regime of a linear network. Now, interesting is when we go to the nonlinear regime, actually, we take the same network essentially, but add a realistic nonlinearity and investigate what we get when we now drive the network with input that is basically matching or is provided by this singular value analysis. And when we do this, then we find that when we drive the network with the leading singular value connectivity modes, then actually we get input that is quite reliable, try to trial. And when we drive it with more higher order singular value modes, we get actually quite high variability between different inputs. And so systematically, this is shown on the bottom here. So actually what we find is that not only is the response of the network highly dependent on whether we drive it with a leading or with a higher order singular value input mode, but also the property that most depended on whether we actually drove the network with an aligned input or not aligned input was actually the try-to-trial variability. So the try-to-trial variability actually depended a lot on whether and how we drove the network, and the mean amplitude didn't change so much, which is actually different from a linear model. In a linear model, you would think that actually when you drive the network with the best modes that you can use, then actually you get the most amplification in terms of activity. Here it's actually try-to-trial variability that was most affected by whether and how you drive the input. Okay. Now, we would try to test this empirically, as I said, and the problem is, of course, in a real cortex, we don't know the connectivity, at least not in ferret. Connectomics is not developed so far that we can take the connectome of the ferret and then drive inputs. But we went one step further with the modeling and making the point that actually we can use spontaneous activity as a proxy for basically for the connectivity modes that give us the most effective drive to the cortex. And so we came up in the model. We used a model for spontaneous activity, basically assuming a uniform random noise. And this showed us that actually the connectivity modes are SVD revealed connectivity modes are very overlapping with the spontaneous activity modes. And so the model suggests then that we can actually use the spontaneous activity principal components, in fact, as a proxy for the main connectivity modes. And the spontaneous activity principal components is something that we can actually access or have access to in experimental data. And we can use this to then test basically whether it is true that in the developing ferret visual cortex, depending on the drive, we get high or low amplification, and whether this amplification is actually mostly affecting response variability and not so much the amplitude. And this is something that is tested in the talk by Gordon. And time is up. So with this, I should come to an end here. I have a few more parts that I will skip, come to the conclusion. So local excitation, lateral inhibition is a powerful network motive for generating model activity. Local self-inhibition makes this mechanism more robust, broadening the parameter regime for model activity. Circuits are heterogeneous. A more realistic model is a perturbed Lili. With heterogeneous Lili, network amplification becomes more input selective and is characterized by high response reliability. I haven't talked about this. I just want to mention this here. We recently published work showing that at sensory onset, actually, the responses are not reliable. And the idea, and just want to sketch this briefly, is that in the light of this work, the possible explanation is that at eye opening, right, the new input that comes from the eyes is basically hitting the cortex for the first time, and they are not yet adjusted to the cortical circuitry, so they come in mostly by random. And so they are not amplified well by the cortex yet. And so because they are not adjusted to like or they are not they're not aligned to the cortical recurrent circuits, and that's why that could explain why you have this high response variability. And with this, I should stop here. Thank people who are involved. Okay.
Thank you very much. All right, questions?
Hi, thank you for the talk. So I'm interested in this heterogeneity. One thing that we know about the cortex is that there are many different excitatory and inhibitory cell types. So I'm wondering first whether you've looked into whether structured heterogeneity as opposed to random heterogeneity might generate other patterns of coupling. And then maybe a second question is whether you can ever observe very highly coupled networks that are not the cortex splitting into two highly coupled networks, but rather three or more.
Into 2 or 3?
3 or more separate highly coupled networks.
So regarding your second question, I'm not sure I'm understanding it. That's why I'm trying to get it right away. So I'm not saying that there are only 2 coupled subnetworks. So usually, I'm also not, so I call it, I wouldn't say these are separate or strictly separated subnetworks. You know? In the sense that it's more, you can think of them as like a relatively lower dimensional set of possible activity states. Yeah, that leads to longer correlations. Yeah, these are not discrete subnetworks, but they are overlapping also in terms of which domains are part of a subnetwork.
So if we think about the homogeneous Lie network,
Yeah.
you imagine that the network connectivity generates a topology that is like a toroidal manifold,
Yeah.
right? But here, what seems to be emerging is that you have maybe two more distant, highly coupled subnetworks, and I'm wondering, is it truly too distant? So, like, have you analyzed the geometry of the new networks?
So when you look at this, so the cloud of activity patterns, yeah, when you sample, let's say, in the model and also actually in the data, then it is on the first, it's actually the first not so bad approximation is like that of a low-dimensional Gaussian. When I say low-dimensional, on the size of this domain, it's like 15-dimensional. Yeah? So, you know, with where the variance components of the Gaussian fall off like exponentially, yeah? Such that when you compute dimensionality like participation ratio, it's on the order of 10, 15, let's say. Yeah? So in that sense, and this is on third approximation, and then you have a little bit of more density, so there is some modulation of this Gaussian. Yeah. So it's more like a very spread out, continuous way. That's also why I'm saying it's not like a discrete set of subnetworks. Yeah. Although it's low dimensional. Yeah. So we haven't really found a toroidal type of geometry here. We have looked into this also in the models, but we haven't found any evidence for that. Yeah? There is also not really a translation invariance here, right, because of the heterogeneity. So that's probably one of the reasons why we don't have that.
Okay, thank you.
Did you have a question? Me or somebody else first? Okay, so first a quick question whether I understood something right. So the beautiful different like direction selective cortical like snaky columns that we all know and love, are those explained by heterogeneous Lie? Because they look a lot like, I mean, so what I was struck by when I first saw the non-hexagonal pattern, that suddenly, more than losing the hexagonal pattern, you got connections between the dots, right? So you kind of get like these snaky shaped columns.
Yeah, yeah.
Is
so
that
the heterogeneity brings it closer to the structure of the orientation preference.
So you think so, yeah?
And then another important factor is where are you related to the critical point, These hexagonal patterns, they occur when you're in the very nonlinear regime, yeah, when really the nonlinearities become important, and they are actually important for determining whether it's a hexagonal pattern or whether it's a stripy pattern, for instance. But when you're only close to to this critical point, but still already start feeling the Lie interaction, then you get a pattern actually that is more still disorganized, has a typical spatial scale, but not really hexagonal structure, and looks in a way sometimes closer to actually the orientation preference map. So somewhere in between, I would say, is the best fit to the experimental data.
Okay, so following that, did you, or maybe I missed it, but connected to what you would predict about the function of the connectivity? So, this goes into this debate of function of cortical columns, and I'm of course thinking immediately like the squirrel monkey, where you have this massive variability and some just don't have them at all, but they seem to just be fine. And so I'm wondering whether you can, is there something that you can get out of, say, a heterogeneous Lie mechanism that would tell you, yeah, you can actually do this to different degrees, and you have this genetically encoded width of the pattern that might be inherited, but it does not actually, but the function is robust to it, basically.
So in terms of the heterogeneity, there's an interesting, I think, link to function in terms of that it produces long-range correlated networks, right? And so when you think of like vision, right? Vision is not detecting like local features, but actually integrating information across space, right? For this, distributed networks correlated over distance would actually make sense. Yeah, that's the heterogeneity. Like the Lie part is less clear, right? And so there is this question, like what is the functional significance of columns which is out there since the discovery of columns like 50 years ago. It's still unresolved. We have actually new data that suggests there is, and I haven't been able to cover this today, but that there is actually suggests that there is a functional significance of columns more in terms of development. Yeah. It seems like it resonates immediately with you, so we should.
Data also, that basically showed you development happens faster and more efficiently if you do have them, but not.
Yeah. So there is something about this so what it does, right? It creates all this correlation, right, between nearby neurons, right? So this reduces the dimensionality of the activity space drastically, right? And so it is the main idea is that it has advantages, right, in development to start with a low-dimensional problem and then increase the dimensionality. So in particular for large brains like primates and carnivores, yeah, this might be a good solution to do. And so I think that this direction is where this could be going.
Yeah, so I have a question actually about the first part of your talk.
Yeah.
With the effective Lilai mechanism. So if you actually step away from this inhibition is infinitely fast relative to excitation limit, are there any dynamical signatures you expect to see of the effect of Lilai via disinhibition compared to the kind of classical Lilai?
Whether I would be able to distinguish this empirically?
Yeah, if you actually have a finite time scale, then you might expect there to be some dynamical differences between those two regimes. I was wondering if you've looked at the finite time scale dynamics and seen anything that might pick those apart.
I mean, nothing that we could test empirically at this point, I think. Yeah. The best way would be to study this empirically would be, I think, patching cells in the paradigital cortex. Yeah. So.
So you mentioned that there's a big variation across mammals, maybe in the scale of the orientation domains.
Yeah.
And so this, you said the idea here is that the pattern formation, the self-organization, yields the structure of these different domains, but their scale is set by the interaction with or some other parameter that presumably is genetically instructed. So is there a genetic correlate to this particular scale variation? Is that something, given the age of transcriptomics now across many different species, that it presumably is something that you can look at by looking at which genes may co-vary with the scale, or something that sets that scale?
Yeah, that's a beautiful suggestion. I haven't done that, but since we have now actually data from, as you say, right, data from on the order of 10 different species that have columns, that could be an interesting, that would be an interesting study to do. And when doing this, there's other structural properties, right, of orientation preference maps and also of other maps that we could then correlate. Yeah, I think that is a beautiful idea. Yeah, I haven't thought about this. Yeah, thanks for the suggestion.
Great, thank you very much.